Good morning or good afternoon, ladies and gentlemen. My name is Helena Kandarova, and I will be sharing today my experiences from the skin irritation validation study in which I participated between the years 2003 to 2006, and that was successfully implemented into the regulations as the OECD test guideline 439. I was involved in quite many successful validation projects. So it was the skin irritation validation study on epiderm, episkin, and SIFT between the years 2003 and 2006, but also catch-up projects either for the skin irritation or for skin corrosion. And later on, I also participated in the Cosmetics Europe validation study for eye irritation endpoint and round robin for the medical devices skin irritation test with the 3D tissue models that resulted into ISO 1099323. And the last project was the retrospective validation of the epiderm phototoxicity test, OECD test guideline 498. I also work as a consultant on several projects that also led to the acceptance of new approach methodologies and technologies into the regulatory framework. So when I will be summarizing some of the key aspects for the validations, my comments will be coming based on all these projects in which I participated. But let's go to the skin irritation validation study. As I mentioned, it was conducted quite some time ago, between 2003 and 2006. And the primary goal of this study was to evaluate whether the epiderm, episkin, and skin integrity function test assays could reliably identify skin irritants labeled as R38 according to the EU risk trace, and the non-irritants, also known as no label, as defined by the European directive. The secondary objective was analysis of the data obtained in this validation study, whether the in vitro tests would be able to discriminate between strong irritants, mild irritants, and non-irritants as defined by the GHS, by the classification and labeling system. You can see how the system was looking in 2003 on the graphic below. Later on, there was a shift for the EU category from 2.0 to 2.3, and that was also retrospectively assessed after the end of the validation study. How and by whom was your validation study organized? So the validation study was quite a large project. The sponsor was ECVAM, the European Center for Validation of Alternative Methods, and coordination was done by a management team. The composition is shown on the next slide. And the contractor had a central role because the organization was responsible for the overall study communication, all the subcontracts, organizing of the meetings, chemical acquisition, organizing study conduct, organizing QC audits, producing interim and final reports, and producing accepted publication. We had 3 test systems in this validation study. It was the episkin test system, the SIFT test systems, and the epiderm test systems. In which of each of the test system was evaluated by 3 laboratories. So overall, it was 9 laboratories participating for the 3 methods. In addition to this, we had independent chemical selection subcommittee and chemical coding and distribution unit that was subcontracted for this purpose. And the biostatistical evaluation was done partly by the ZBET and partly in cooperation with the ECVAM, mostly for the final analysis of the data after the decoding. The study also had US observers from ECVAM NICETAM, and overall it was a project that, despite it was quite complex, didn't really requested so many meetings. In this project, we started in November 2003 by the discussion and approval of the project plan that was drafted by the colleagues from the BFR. And during the course of the study of the project, we had 6 face-to-face management team meetings and 6 management team teleconferences. If you are interested in more details about how the study was organized, you can find it in the publication of Professor Spielmann from 2007, published in ATLA. So you can see on the next slide the management team composition. You most likely know all these names. These people are people who had a lot of experience from the previous pre-validation project and the validation project that was running for the skin corrosion endpoint, experienced biostatisticians, and also the observers from the United States had a lot of experience on previously conducted validation studies. I think that the success of the validation was partly from my point of view, also due to the excellent management team that took care for conductance of all the steps in the projects as they were defined by the project goals. How and by whom was your validation study funded? So, validation study was funded by ECVAM. It was in 2003 in June when the European Commission issued a call for tenders for the skin irritation validation study, and the Federal Institute for Risk Assessment submitted their offer in July 2003 and received the management contract in November 2003. The maximum total amount paid by the Commission under the contract was €858,000, and it was covering all tasks executed for all 3 test systems, including the biostatistical report. You can see the breakdown of the costs. So the management was basically calculating with 2 scientists, and quality assurance audits. It was in the proposal marked as a package 7. Overall cost was 132,000. The finances needed to validate the test systems were around €200,000. Test system 1 was 200,000, test system 2 was 168, and test system 3 was 225,000 euro. Meeting costs overall, they were very, I would say, modest, 53,000 euro. Chemical purchase, coding and distribution, that was 55,000 euro, and production of the statistical report under the package 6 was 25,000 euro. In the following slides, I give you some details, cost breakdown for the different packages. I will not go very much into the details, but you can see that all the costs were very precisely calculated, and they really thought about all possible steps that needed to be conducted and also budgeted for the successful validation project. Laboratory management design. How many laboratories were involved and whether they tested all the chemicals. So the project counted initially with 9 laboratories to be involved in the validation study. In phase 1, 3 laboratories conducted the testing. It was L'Oreal, Syngenta, and Zibet. And in phase 2, we had only 7 laboratories because phase 2 was not conducted for the SIFT test. It was for the reason that in the phase 1, SIFT didn't provide satisfactory outcomes with regard to the sensitivity, and the decision was not to proceed into the phase 2. The phase 1 laboratories, they were also responsible for the trainings of the another 2 laboratories for their test systems. So they had a lot of responsibility for their own test assay, for drafting SOPs, methods documentation sheets, and they were assuring that the method is correctly transferred to the other 2 labs participating in phase 2. All laboratories tested all chemicals. In phase 1, we had 20 coded samples, 9 irritants, and 11 non-irritants. Each chemical was tested in 3 independent runs in 3 laboratories. In phase 2, we had 58 coded test materials, and there were 18 chemicals that were the same as in phase 1 to check the intra-laboratory reproducibility. And additionally, we had 40 new chemicals that were selected from the new chemical database and some existing sources, databases like the ACETOC list. Each chemical was tested with the MTT test in each laboratory on 3 parallel tissue replicates per test in 3 independent runs. And the test samples were frozen, the media, the samples were frozen to allow for the subsequent determination of the interleukin-1 alpha as additional endpoint. The number of the chemicals required for this validation exercise was determined statistically by the sample size calculation. If you are interested about the details on the chemical selection and acquisition and some other details, you can find these details in the publication of Chandra Eskas, also published in ATLA in 2007. How was the selection and procurement of the reference chemicals organized? Criteria, supplier, shipment, coding, and decoding. So the selection and procurement of the materials was done by the independent chemical selection subcommittee. These were people with a lot of knowledge in toxicology and chemistry. The chair of the committee was Dr. Andrew Worth. We had representatives from ECVAM, Dr. Chandra Eskes, and Dr. Varai Zhuang, representative from the ECB, Dr. Tom Cole, and representatives from the competent authorities. From the BFR, it was Dr. Ingrid Gerner, and from the HSC UK, it was Dr. Amanda Cockshott. Criteria influencing the selection. So there was one problem in the validation project, and that was the fact that most of the materials that were described in the ECTOC database were basically the in vivo data were published. They were used in the development and optimization of the method. So there were not many materials left that could be used for the validation projects. That's why the chemical selection committee, together with the management, decided to use the chemicals from the new chemical database of the European Chemical Bureau, where high quality in vivo data were available. There were a number of the exclusion criteria that were followed, and they are in detail described in the publications of Dr. Eskes. I will mention a few. Substances that were notified before 1995 were initially excluded because it was a bit complicated to find all the details that were needed basically for the project to validate the test methods. The repeated notifications were excluded for multiple dossiers concerning the same substance, and only data from the file leader, usually the original notifier, were extracted and used. Substances marked at quantities below 0.1 ton per year, for which skin irritation testing is not a regulatory requirement, were excluded. Skin corrosives were also excluded, as the primary focus was skin irritation endpoint. Absorbents, gases, and vapors were also excluded for the technical reasons, because the protocols were not adjusted to test this type of the materials. When the chemical selection committee applied all these rules to the new chemical database, there were not so many materials remaining, and additionally, when secondary data reduction rules were applied, there were not really many materials left that could be tested. So in total, the selection committee ended up with some 126 substances that were selected as suitable materials for the validation purposes, and they comprised of 32 skin irritants and 94 non-irritants. Further on, when the chemical selection committee and the contractor started to discuss the purchase and supply, the responses were received from 30 companies, of which 25 were able to cooperate. You can see the names of the companies and the locations of the companies in the table 2 from the publication of Eskes et al. And finally, only 33 materials from the new chemical database were finally available for the study. So due to the lack of the substances from the new chemical database, some additional materials had to be used from the existing sources, existing substances, with data resources from ECTOC and TSCA. So it was quite challenging for the chemical selection committee, as you can see. Coding, decoding, and transport, that was done by an affiliated intermediary. It was the research and consulting company Cytotest Research, Rostov, Germany. They took care for the coding procedures that were prescribed by the BFR. It was done in a very good quality. Samples were sent within the European Union and also to United States via the expedited couriers. All the samples were coded, they were sent coded, and safety data sheets were supplied in seal enveloped, and they were provided to the safety officers of the testing facilities. The envelope could be opened only in case of an accident, and they had to be returned back after the finishing of the validation trial. There were some additional administrative steps needed for the samples that were sent to United States, but finally we succeeded, and also for United States the samples were delivered in good shape and delivered coded. Samples were transported in dark, high-quality borosilicate glass. The only drawback was that the subcontracted company selected vials with the rubber septum instead of chemically resistant Teflon. And this error caused that some of the materials reacted with the septum, and consequently we thought that these reactions could have been modifying also some of the irritating properties of some of these materials. We, however, carefully collected these observations and reported them to the management. So when the materials were decoded, the management team had information on this reactivity back to in vivo and in vitro data. What were the main organizational challenges? The study was really extremely well managed and conducted in laboratories with previous knowledge on how to run the validation projects in a GLP compliant mode. Thus, there were no really major surprises or problems regarding the organization challenges. All roles were precisely defined in the study plan and agreed before the start of the validation, and there were no modification needed. There were small challenges that I mentioned. That was, as said already, supply of the coded materials to United States, but there was a specialized company that was able to conduct this task, and the samples were delivered successfully. There were lack of the suitable chemicals with a high quality and publicly available in vivo data and physical chemical properties. Thus, in addition to the samples selected from the new chemical database, we also selected some of the materials that were provided from other databases. And the purchase of chemicals that were selected from new chemical database, that was a bit challenging. The communication took longer than expected, and some of the materials were too pricey, so we could not really order everything what the initially suggested the chemical selection committee. There were also some minor challenges. There was a visa problem for one of the two auditors, which was me, so I couldn't conduct the audit in the US facility, and we needed to train very quickly another auditor. And for the technical problems, some of the materials that were selected had a really bad smell, and so we needed to install some additional chemical exhaust in these laboratories and over the laminar hood. So that was basically it. What were the main financial challenges? What solutions did you find? So the study was initially really carefully and realistically budgeted, so there were no significant problems with the funding. And additionally, since the SIFT test didn't proceed into the phase 2 of the validation, the SIFT budget could be used to analyze the culture media samples for the content of interleukin-1 alpha, which was suggested as a secondary endpoint. In conclusion, to what factors do you attribute the success of your validation study? I think there were several factors that contributed to this success. At the project management level, it was the professionalism and the experience of the project management team who dedicated their time fully to the project. That's also important, not only experience, but also the time that the people dedicate to conduct the project is very important. We had very clear goals with strictly defined aims. The project plan was drafted, discussed, and approved by all parties before the start of the study, and the management team took care that the project was precisely executed, so everyone was clear about what to do, how to do, and when to do. On the technical levels, of a benefit was that participating laboratories had already experience with validation trials. They were basically prepared to do the work that was requested from them after very short training. The technology was ready, and in addition, um, we conducted the quality assurance audits to be sure that everything is prepared for the validation project and that there were no surprises whatsoever from the side of the skin model producers. And overall, also, I need to say that the documentation and reporting was on very high level, and it was not a GLP study, but it was a GLP compliant project. On the financial level, it was very good that we had realistic budget. The management team and the laboratories were very careful in spending the money. There was a big preference to hold teleconferences over in-person meetings. And as I said already, it also helped that we had experience and already well-trained partners for the project conductance. Space for improvement. So I summarized what I learned in the validation studies and I have seen later on in some of the projects where I was involved. I think that it's very important to seek for the validation projects organizations and experts who have already experience with validations and validation standards. It's also very important to count with more time for the chemical selection, for the purchase, and for the distribution. We need to also be very clear about what is our standard, gold standard for the data that we are comparing to. And I do think that whenever human data are available, they should be considered when discussing the outcomes, especially for the inconsistently predicted materials. It is very important to hire experienced biostatistician who could help not only at the end to evaluate the data, but also at the beginning to calculate the needed sample size and consider the prevalence of the effect. I think one of the very essential things is that we need to be clear about the validation goal and the aims of the study. That should not be changed during the conductance of the study. At the technical level, in general, every system that is undergoing validation study should have, in my opinion, certain level of the technology readiness. Ideally, at least these systems should be partly commercialized. It's very risky to start to validate something that is in the stage of the prototype because we cannot really do validation and after the validation to change the model that basically disvalidates the whole effort. It is impossible to fine-tune the protocol or prediction model in between the validation phases that I have also seen some attempts for this in some of the projects. It is important to implement the GMP and GLP audits at the developer side before embarking on the validation. And laboratories or the management especially should insist on the training and transferability exercise before starting the main validation trial. I still would like to mention that the pre-validation concept is really valuable and can save a lot of time and the money. Not everything should be called validation. Validation has certain aspects and certain things that are called validations nowadays are not validations. They are basically pre-validations checks of the technology. At the financial level, I think it's very important to have realistic budget done by experienced professionals, involvement of experienced and already well-trained partners that are fully committed to the validation and they will not that will not put the validation on the second or third level of the importance within the company. And of course, careful spending, that's key to the success whenever possible. Teleconferences over the in-person meetings, they can save a lot of money. Where to find money for the validation study? So I would like to mention that the historical reason for founding ECVAM was funding and management of validation studies and the support to other commission services on issues related to alternatives to animal testing. So I think that we should again see EU-sponsored validation tenders that should be conducted under the ECVAM leadership or in involvement, direct involvement, I think. And of course, there are usually other projects running in parallel, public-private projects that could be also very valuable source for additional funds for validation. Member states should also support their NETFAL laboratories by fixed amount budgets that will allow them to conduct these type of the projects. And I think that we will also need to improve significantly the infrastructure in the laboratories that are conducting these validation projects, because some advanced systems are very pricey and they are not readily available in the labs. And I do think that the scientific and validation potential of the EU NETFAL laboratories is not yet fully used, and with their contribution, the validation studies could run faster and could be at the end also cheaper. I would like to acknowledge help of my two former colleagues, Professor Spielmann and Dr. Manfred Liebsch, in preparation of this presentation. And if you would like to contact me for some additional questions, please don't hesitate to send me an email to this email address below. Thank you very much for your attention.